Hi, my name is Shane Kozick. The brain surface is made up of sulci and gyri that together create the distinct folded sulcogyral appearance of the brain. The orbital frontal cortex, abbreviated OFC, is one brain region with a great deal of individual differences in sulcogyral variability. Specifically, the H sulcus, which is formed by the intersection of the medial, lateral, and transverse orbital sulci within the OFC, has been a particular focus of ongoing research due to its potential as a morphological mark for various psychiatric disorders. Figure 1 depicts cartoon illustrations of the brain that show the four pattern types identified based on the continuity and discontinuity of the red traced MOS and the blue traced LOS. These patterns are named according to frequency in the general population, with type 1 identified in 60%, type 2 in 25%, type 3 in 10%, and type 4 in 5% across studies. It has been found that the spatial organization of these patterns are not identical in every individual that has the same overall pattern type. Rather, there are subtypes that can also be characterized within each pattern type. Subtypes offer a more fine-grained characterization of the OFC. We were interested in expanding our understanding of OFC sulcogyral development within a twin population of neurotypical individuals. We examined whether there is an increased concordance of patterns of 172 monozygotic twins relative to 304 dizygotic twins. We defined concordance as the twins having identical pattern types on both hemispheres of the brain. We then expected monozygotic twins to have more similar subtypes if orbital frontal sulcogyral patterns have a strong genetic component to them. Moving on to the methods, we use publicly available data from the Human Connectome Project for monozygotic and dizygotic twin MRI scans that are processed through an automated tracing pipeline. This pipeline was used to identify the sulci making up the H sulcus. Anatomist, which is a neuroimaging software platform, was used to manually label the individual sulci fragments on both hemispheres of the brain. The initial pattern type was predetermined, and the subtype was then characterized based on the orientation of the MOS, TOS, LOS, and IOS by two independent researchers. Figure 4 depicts 14 cartoon trace subtypes that exist within type 1 pattern. All the subtypes are characterized as type 1 because they have a discontinuous MOS and a continuous LOS. However, the orientation of the other sulci within the subtypes differ with respect to each other. We identified a large number of pattern subtypes that have not previously been described in this study. We continue to identify new subtypes that don't fit into the initial subtypes described in the seminal paper. Another way we could understand these subtypes is through principal component analysis, or PCA. Distinct features of each pattern type were described, and then each subtype was coded and given a unique vector of answers of two questions we asked. Examples of these features are, does the rostral MOS intersect with the IOS, and does the MOS have multiple branches? In the radial plot, the rings represent frequencies of each subtype. Overall, these results suggest minimal genetic influence on OFC pattern types, since we do not find an overall higher concordance of pattern types in monozygotic twins. Given that atypical pattern types have been associated with psychiatric disorders, these results suggest that OFC sulcogyral patterns capture important variants that is not genetic in origin, but may be represent to psychiatric disease risk. Analysis with pattern subtypes are ongoing.
No.